Hey everyone, I'm going to show you how to use DSViewer in this video tutorial information for you to make your figures for the paper. So to get started, if you go to Angel, you'll see I have the user's guide, the structural biology lab folder that contains the paper for the HIV protease, the TATA binding protein. And as I said in lab, you can do your own protein if you want. Just make sure it has a substrate or a target, something else that interacts with a protein like actin wouldn't be necessarily a good protein to do because it doesn't do. There's going to be very little to show. Basically, what's right here, actually, you know what? I'm going to open it up and just point this out. At the bottom of this one, there are some suggestions for the TATA binding protein with different files you might want to use, like the 1TGH file. But I'll show you how else you can do that. So if everyone goes to Google and types in PDB, they'll bring up the protein data bank. That should be your first link, the RCSB protein data bank. That's the one you want to click on. If you click on that, and for us, let's choose the HIV protease. HIV protease. Type that into the search bar. You get a whole bunch. You get a whole mess of proteins that come up. I might want my X-ray resolution to be fairly small, meaning that like they know where the molecules are within just a couple, angstroms, 1.5 angstroms. Sounds good to me. All right. So if I look at these, some of these are complex with different inhibitors. Doesn't really matter which one I choose. They should all be fine. I'm going to choose this 4HDF one. So if you open up Discovery Studio, it looks like a white square split, or it looks like a square split to a white and blue pieces. I'm using Discovery Studio 3.5. If you're on the labs in the computer, you'll be using Discovery Studio 2.5. There are some slight differences, but I will talk about what those are when I go through this. So what you want to do is you want to go to File, Open URL. And if you don't remember what URL we're using, it's this 4-digit code that you see here. So, well, you'll want to find a paper that's a good paper. I'm just gonna pick this first one that comes up. Okay, 1HXW. Hopefully this is a decent one to just show you how to do this stuff on. The protein you pick, it will be based on whether the resolution is high, whether there is a inhibitor or not. You'll see why in a second. The other things you want to consider is the primary literature article that this came from, like the people who did the X-ray crystallography. You'll see that in the journals and the authors and the PubMed abstract here, which allows you to go get access to that paper. You probably want to pick a structure from a paper that you're reading. So, for example, this HIV protease paper, you might want to look at the ones in that paper, but since that paper is a little older, you might want to look for something that was published more recently. This one will do for my example, or it should do for my example. So this 4-digit code, that's the URL I want to open, that's the PDB ID number, so I type that in here, hit open. If this doesn't open right away, what you can do is you can go back here and you can go to download files, and you can you can highlight download. You want to download it as a PDB file, and you'll save that to your desktop, and then in DSViewer you can go to File, Open, and find that file. Now what I have here is slightly different than what you have if you're using version 2.5. Mine is already colored by secondary structure, and you can see all my little water molecules around this. What everyone wants to do, whatever version you're using, is hit Control-H. That'll open up your dialog window. Then turn off the water. You don't need all that water. Click on the 1HXW. This will highlight everything, and then go to Display Style. So for me, Display Style is in this nice box that says Display Style. In 2.5, it's a small square, probably more over in this area, and there's a red, green, and blue dot followed by some lines on the side. If you just mouse over it, the words display style will come up. Click on that button. Click on that button. You're going to have atom type. You're going to have a couple tabs here. You're going to have atom type, protein, graphics, lighting, materials. Go to atom type. Select none. Hit apply. Go to protein. Select. Here's where you can select a couple different things. CA wire. If I hit apply, this is not the best way to represent your data, but if you're superimposing a bunch of proteins on top of each other, that's a good one to use. CA stick isn't much better, just like thicker wire. Line ribbon, this one can be kind of cool, but I don't think it's the best way to represent your data. Flat ribbon's fine. Solid ribbon is my favorite because it looks like it actually has some, I don't know. It looks like there's something there. You know, it looks like it has 3 dimensions to it. Then there's tube, which is kind of like CA wire or CA stick. This can be important if you're showing some turn, because this shows the direction of the backbone of the protein. Then there's schematic, which can show you the alpha helices, the beta sheets. It's kind of nice that way. But if you go to solid ribbon, you can also express that by coloring by secondary type. So right now, all of you were probably colored by residue. Let me show you what that looks like. So yours probably looks something like this. But if you go back in, you click on protein, and you color by secondary type and hit apply. The red represents alpha helices. The blue represents beta sheets. I believe the green is just linker proteins, so it's connecting this beta sheet to this beta sheet, this alpha helice to this either N or C terminus of the protein. You'll notice there's not that molecule that was in the center. That's because I turned atom type off. We want to turn that back on for that one that was in the center. So let's try to figure out which one that was. A in our dialog box represents this half of the protein. You can see the HIV protease is actually a dimer. So if I were to shut this protein off, which I can do by unchecking the box, and I spin this around, you can see this looks exactly the same as the piece that I just lost. If I turn it back on, you can see both sides are the same. So it's a dimer. B. Hey guys, I'm interrupting this video with this because I realized I left out something for those of you who are doing the TATA binding protein. If you've chosen the TATA binding protein, eventually you're going to get an image that looks like this. As this, you're going to click on the 4-digit code, and you're going to go to display style. And for you guys, I'm just going to do this really quick. You guys probably have something that looks more like this, just like a mess. What you guys will do is you'll open the display protein, you'll turn atoms off, you'll turn the protein on, and you'll want a solid ribbon. And then you'll click on this tab, the DNA RNA. What you want to select is you want to select arrows, and you want to select a ladder. If you hit apply, that's going to make it look a lot nicer. And then if you're in 2.5, it's colored by residue right now. You want to go in and you want to change that to secondary type. Apply, okay, and you should have something that looks like this. Alright, back to HIV protease. Let's do CPK. Okay, yeah, so I can click CPK to show this. I could click line. This is the inhibitor of this protein. There's a couple different ways you can display atom, stick, ball and stick. Ball and stick's probably the best, or scaled ball and stick. CPK is a space filling model that also shows charge. I don't know what polyhedron looks like. I don't like that. Okay, you know what? Let's leave it as CPK for a second to show you what CPK does. So you can see this shows you how much space that's actually taking up. You can do this for individual proteins as well. For example, if I select this protein and I go to display style under atom type, I can show it as line, and this will show the R group coming off that protein. Or I can show it as stick, you know, scaled ball and stick. That's typically what I choose to display them as. None though, because that one's not particularly important. the R groups that you're gonna want to show are R groups that maybe interact with this inhibitor. And the way the inhibitor is kind of working, as you'll read the paper, the beta sheet flaps up on top. Basically, because it is a protease, it's cutting protein. These two flaps, they act to come down on a protein chain. This is mimicking that, but it has different bonds that prevent basically the cleavage. I think these two arms fold down and grab this inhibitor as though they're going to cut it because they're recognizing it as a protein, and then they can't. But we need to identify the important amino acids that are interacting with this. The nice thing about the HIV protease, and I don't know if you guys have this, I'm going to assume that all of these are important in the role because they're under this heading active site proteins. I don't want you guys to assume that. Anything you write, you need to prove. For example, let me just say this. I one time, if I remove this, I one time said, when I look at this protein, I kind of see a dog face. I see the nose here, I see whiskers here, I just kind of see eyes here and ears here. Now I know that's stupid, but whatever. I had a student write that the HIV protease, she said, the HIV protease takes the form of a dog or something. Before you go and say, you know, this protein is important for binding the proteins, verify that through some form of primary literature, not just because I said so, okay? Alright, so moving on. Whoops. Alright, back to what I wanted to do. If I click on active sites, I can display them as ball and stick, and I go to structure and monitor. I can monitor any hydrogen bonds that this amino acid has. Okay, there. I found some that have hydrogen bonds. What that's going to do, and you'll know that it has a hydrogen bond based on your reading in the primary literature. And then you'll be able to select that amino acid. So if you know that it's an arginine and it's the, I don't know, 75th amino acid or something, you can go to your hierarchy window, and if I do that and I click on and select one of the dimers, I can click the drop down box and it gives me a breakdown of the amino acids. At the first amino acid, it's a proline, and then you can see it just keeps going up 2, 3, 4, 5. In your paper, it'll tell you, you know, that valine 82 is important for whatever reason, and then you can click on that in the dialog box, and you can go to display. You can choose to display it as ball and stick, and you can look to see if it has any hydrogen bond interactions by going to structure, monitor hydrogen bonds. Alright, so let's say that valine at 82, which does not actually have any hydrogen bonds, let's say it's something I want to label. I can go to structure, I can go to label, I can go to add, and then I don't want to label it as an atom, I want to label it as an amino acid. And I want the color to be something that isn't like any of the other colors in this. I might go with a pink. Okay. And then I'm going to make it big. Okay, there. And then this shows up right next to that that molecule. Another thing you can do is you can, if you get your picture and you like how it's set up, but you're having a hard time getting your labels to display where you want them to, is you can save your picture first and then use something like Photoshop or even PowerPoint to overlay your titles on top. And then you'll have a little more control where they go. But yeah, so now let's say I wanted to color all those active sites. And for me, I have this group in this hierarchy window that highlights all the active sites. Let's say I want to make these all some color that really stands out so that I can say all the important amino acids that are that play a part in the active site are colored, I don't know, they're colored, They colored this sort of light red. Okay, apply. There. Something that's different enough from the red of the alpha helices, and it's not blue or this teal color of the beta sheets. If the whole point of this picture right now was to show these hydrogen bonds down here, this picture would be a mess. If I wanted to show that, I might want to take away, I might want to remove, yeah, a whole subunit even, just so I could get in and, you know, get up close to whatever it is I was trying to show or display. Another thing is you might find that something like this amino acid is in the way, so you could choose to go to atom type none and just remove it. If you knew the actual amino acid, maybe it was something like this right here. When you highlight an amino acid, it highlights whichever one in the window it's under, and that apparently right there is a leucine. So I could like deselect that and remove part of this chain, so I could really get in here to look at this hydrogen bond, for example. Now, remember, if you're in super close like this, this doesn't give me context for where I'm looking at. So you might want to have a picture like this, you know, nice and zoomed in, and then zoom way out, you know, and show me what the whole structure looks like, and then maybe just show me a picture of what this looks like down in the corner, so I know what you're looking at super zoomed in. That's just an idea for you guys. You might want to add a surface to your protein, but still want to see the underlying structure. So I told you how CPK gives you this space-filling model for your ligand in the middle, and you don't have to represent your ligand, or inhibitor, I mean, through that CPK. You could do it ball and stick, that's fine. You know, that might actually be a little clearer when you're trying to show all these hydrogen bonds. But the other thing you might want to do is you might want to show how little space there actually is present here. So what you can do is you can go to your protein, and you can go to structure, and you can go to surface, and then add. If you add solvent, this will show you basically what is actually exposed, what space there actually is. And what I like to do is I like to turn the transparency way down so that you can actually see the ribbons underneath to know what the secondary structures are that you're looking at. So if I do that and I click OK, this is very similar to CPK. You can see the blue and the red indicating charge. And I'm going to undo this. You can Control Z just like you do. Oops. Yeah, just like you do in Word, but it's going to undo every single rotation that you've done. What I want to do is I only want to add solvent for A and B. So I'm going to select A and B. And then I'm going to go to Structure, Surface, Add, Solvent. Okay. Okay. Now you can see it left this hole in there. Now you might want to leave it just like that hole, but this is where it might be a good opportunity for you to use CPK on your inhibitor. So if I click on B, which highlights the inhibitor, I can go to Display Style, I can change that to CPK, hit Apply, hit OK. And there, now you actually see how tightly that fits in there. It engineered this inhibitor to just perfectly, you know, fit with this HIV protease. You might want this to be a little more clear, so you might want to color this one color. You can go to Display Style. You can choose maybe a real dark blue, something that contrasts with everything else. Okay, yeah, okay. You know, maybe dark blue isn't the best option. I don't know. That's up to you. But I think I've given you most of what you're going to need to be able to do this. If there is a specific thing that you want to copy to a new window to look at, you can go to File, and you can click New Molecular Window. You go here, If for some reason, I don't know, the amino acid 10 through 71 is super important to you, you can hit Control C, then you can go to your new window and hit Control V and just work with this part of your protein. That might be useful for some people looking at certain things. Play around with that if you like. No. Now, let me show you one last thing using PDB. You can do these things like protein overlays. So, if I go back in here and I type HIV, let's say I do want something new. Okay, 3ZPS. Let's say I care about this one for some reason. If I go over here on the side where it says Tools, I can click on Compare Structures. Type in 3ZPS. And I'm going to choose Part A, which is just half the dimer. Okay? And then I want to select Fat Cat Rigid 4AG9. ZPS. Okay. So now I'm comparing 4AG9A to 3ZPSA. I clicked yes for the alignment. I'm going to go down to here where it says download PDB file. I'm going to right click and I'm going to save this link as alignment. In here, I'm going to hit File, Open. And I'm going to scroll through my desktop, click on alignment, hit Open. That's going to open a new window that has this alignment. I'm going to open my Control H, open my hierarchy window. You can see that they're right on top of each other here. You can see how they're right on top of each other. They're very similar to one another, except for up here. There's a slight difference in this loop here. So one of these ends is the C-terminus, one is the N-terminus, but you have these two proteins overlaid on top of one another. So if there's different inhibitors you can use that tool perhaps to see what the difference is or how similar some of the structures are, between old and new. It's a cool tool. I didn't use it when I did mine, and most people haven't used it, but if you're looking to make kind of a cool structure, you might want to look into this. I think that's basically everything I want to show you. Let me just show you how to save now. So I want to go to File, I want to go Save As. I want to make sure I save it as a Discovery Studio file, because this will allow me to come back in and all my work will still be here if I save it as this. So I'm going to hit Save. Okay. The next thing I want to do, if I want to actually save this as a picture, is I want to have it set exactly as I want it, because the second I change the quality of this picture to high, working with it becomes incredibly hard on your computer and difficult to do. It'll take forever. But yeah, so let's say this is the molecule I want a picture of. This would be a terrible figure for you guys. Like, this shows me there's way too much going on in this. You want to focus on smaller things. But just for an example, what I would do is, so you can just click display style, doesn't matter what you're looking at, and you go, okay, I was working at high. Most of the time you want to be working at medium or low, just so that you don't put strain on your computer. But I'm going to go to ultra high, and I'm going to change the background image color to white. Hit OK. You see this little box called image next to the background color? That's because you could literally upload an image of even yourself holding, like, maybe your hand out so it looks like you're holding your protein. That'd be kind of cool. But otherwise we want to go with white because we don't want to waste all that toner on just this black background. Then I'm going to go to lighting because I think my lighting could look nicer. I can add a light in, and I can move the lights around to see what they'll look like. If I hit apply, all of this is going to apply. And it's going to take a minute because I should have been doing this, step by step. But here we go. Oh, display not responding. Oh, okay, hey, cool. So, You can move the lights around, hit apply, figure out what looks good, apply. You know what? That looks fine. Hit OK. There. Now, like I said, I do not want to try to move this. It should be set by the time you're doing that. Then you want to go to File. You want to go to Save As. You want to choose. You can leave the same file name. Just choose image files under the Save As type. Click on Image Files. For you guys, it'll probably say JPEG, PNG, whatever. But the first part, it should still say image files. So save it as an image file. Save. And this part, the units should be pixels and aspect ratio should be logged, so this box should be checked. And then you want to change either width or height. I'm not sure which one. It won't matter. You're going to get a high resolution image, but I'm pretty sure it says in Malachi's directions. You can look. You want to change that to 2000, and don't worry about the other box. That will change automatically. If you watch for a second while I hold OK, that changed. It changed to 1240. So I hit OK. If I go to my desktop, I should be able to see it. Yep, it's right here. And I have a nice image. What the nice thing about having it ultra high and at such a big picture like this is when you shrink it down to put it in your appendix for your figures, it'll just have this nice glossy look. That's how you make an image. I know this was long, but hopefully this helped. If you're at this point, you know how long this went, and you also know that pretty much all I did was go over what is in Malachi's handout. So, yeah, those are both tools for you to use. If you have any questions, email me or Dr. Fountain. All right, how do I shut this off?